So this is continuing on with our land theme. We've got Will Wieder, who's the current land model working group chair. The one of two positions. Who's the other? Rosie. Oh, Rosie Fisher. Okay, so Rosie Fisher is our other external chair, sort of external. But so Will's going to give us an overview and an insight into biogeochemistry in the land model. And I'll hand it over to Will.
Great, thanks. Part of this is sales pitch and part of this is educational. So I'm curious how many folks here would consider themselves biogeochemists? Oh my gosh, there's like 4 of you. Yes. All right. And so everyone else, you do something else, and it's not biogeochemistry, and it's probably not land biogeochemistry. So part of my job is to get you excited about what we're trying to do in the land model and what we're doing in CESM with biogeochemistry. And for CESM, biogeochemistry is really the carbon cycle. And for the oceans, they think about like the whole periodic table of elements and how silicon and iron and phosphorus and nitrogen all affect the carbon cycle. On the land side, it's mostly nitrogen is the other element that we're concerned about. So if you're here to hear a lot about nitrogen, sorry, it's mostly going to be a carbon talk and carbon on land. How do I do this? Oh, there it goes. Okay. You guys are doing stuff too back there. That's exciting. Okay, so in terms of climate change prediction and attribution, I think we're making huge strides in the physical climate system and being able to attribute individual events like the heat dome that hit the Northwest last year, the extreme heat that hit England just a month ago, and then this past spring. You know, the attribution studies on the physical climate side are getting really advanced, and so we're able to attribute the likelihood of individual events and their role in climate change. For the carbon cycle, though, how do I? Sorry, I'm a rookie here, guys. Wait, no. There, cool. Is there something I should be pushing? The arrows. Ah, not the mouse. Got it. Okay. So in terms of ecology and understanding the ecological impacts of climate change, though, I think we're maybe a decade behind. It's much harder for us to understand how extreme events are impacting ecosystems and how those impacts on ecosystems are affecting the carbon cycle. And so that's one of the, I think, the big challenges that we have in thinking about biogeochemistry, largely in CESM and especially in the land model. So I'll talk some about why that is. So the first part of this talk is why even worry about biogeochemistry and CESM. Second part is how do we actually do it? The nuts and bolts are too involved for a 40-minute talk. So this is going to be necessarily brief. And then finally, kind of how are we doing? What are the uncertainties that we're still facing with? And then what are some of the future directions? Okay, so for this first part, why? Why do we worry about the carbon cycle in CESM? So if you're a land person, if you're a terrestrial person, and you study lakes, you study glaciers, agriculture, if you're an oceanographer and you study, coral or fish or anything human related in the sea, or if you study the built ecosystem in urban ecosystems, it's obvious that ecosystems are affected by climate change. And kind of my smug answer is why I study the carbon cycle is, well, you can't eat the Madden-Julian oscillation. Like it's not very tasty. It's not very delicious. It might fill your mental soul, but it doesn't fill your physical soul. So it's really important to study the carbon cycle, especially on land, because that's what we eat. The other reason to do it is because of this figure. So this is the Mauna Loa Observatory looking at CO2 in the atmosphere from 1960. And what do you guys notice? It's going up. What else do you notice? What are the wiggles? Yep, yeah. So those wiggles are the imprint of land on the atmospheric record of CO2. And so when it goes up, that's winter in the Northern Hemisphere and CO2 accumulates at a faster rate. And when it comes down, that's summer in the Northern Hemisphere and it's the biosphere breathing. So every winter it exhales and every summer it inhales. And you can see that in the atmospheric CO2 record, which I think is fascinating, that you can look at the imprint of land and we'll get a little bit into there was a question just before the break about interannual variability. So you can actually see interannual variability in the land carbon signature in the atmosphere. So it's a big deal. And the whole reason that we have Earth system models, in my mind, is because of this figure. Like, it's great to learn about atmospheric dynamics and ocean circulation, but none of it matters if you don't put this on top of it. Yeah, did you have a question? No, there actually is modulation over time. You just can't see it very well the way this is displayed. So there is interannual variability, but you can't, it's hard to see here. No, actually the amplitude of the peak to trough is getting bigger over time. Yeah. Yeah, so the amplitude of the seasonal cycle is what's very variable, but it also, depending on where you look, and it's harder to see in the Mauna Loa record, but at Barrow you can see it more strongly. So at higher latitudes, you see that amplification more obviously. Yeah, good questions. Please interrupt. If I talk for 45 minutes, you guys are gonna get really sick of hearing my voice. So I would love questions, and shout them out if I can't see you, if you're back in the corner. Okay, so biogeochemistry and CESM is all about the carbon cycle, and it's all about trying to understand the historical CO2 record, as well as make projections about it in the future. So in my mind, it's super, super important. Here's what that looks like in terms of the fossil fuels that we've emitted. Oh, I don't have my notes on these slides. That's unfortunate. I'm going to try to do this by memory. So something like 660 petagrams of carbon have been spewed into the atmosphere, and roughly a third of that is actually from land use and land cover change. So, when you think about climate change, you think about CO2 in the atmosphere, roughly 450 petagrams is coming from fossil fuel emissions. That's the gray bar here. And roughly 200 petagrams is coming from land use and land cover change. That's the yellow bar here. Sorry, I don't know where to point. On this figure, on the cumulative emissions. On an annual time sense, early on in this record, especially before 1920, 1940, again, it's mostly land use, land cover change that's causing the emissions. Later in the 20th century or first part of the 21st century, it's all about fossil fuels. So it's all about CO2 from fossil fuel emissions. Where's it going? I blotted out the bottom of this figure. So we've thrown up 660 petagrams of CO2 into the atmosphere. Is it all there? Ocean? Where else is it? Atmosphere and land. There's a lot of it on land. Okay, here's what it looks like when you show it. So the dark green, these colors are not very good. So this dark green, this is oceans. Oceans have taken up 175 petagrams of carbon. So roughly a quarter has gone into the oceans. The residual CO2 in the atmosphere is this blue bar. What's left over then is land. So the green bar, 250 petagrams, more or less. There's a little bit of an imbalance in this bookkeeping method of about, I think it's 5 to 10 petagrams. Anyway, so of all the CO2 we put in the atmosphere, a quarter of it's in the ocean, 30% of it actually is on land, and the residual, about 40%, is in the atmosphere. So to me, that's a huge ecosystem service. Oceans are providing a massive ecosystem service by sponging up CO2, and land is also providing a massive ecosystem service. So in some ways, land vis-a-vis humans is part of the problem in terms of land use change, but it's also part of some of the solution in terms of sucking CO2 out of the atmosphere. You can see the wiggles here really strongly of that interannual variability. And so the interannual variability of CO2 in the atmosphere is evident by these wiggles here. Most of that's driven by land. And so it's interannual variability and land carbon uptake that's driving this interannual variability of the residual CO2 that's left in the atmosphere. These are results from the Global Carbon Cycle project that came out last year. And that's highlighting the interannual variability. Okay, so that's in reality. We kind of have a good idea, based on this bookkeeping method of knowing how much fossil fuel emissions have been there, getting a pretty good estimate of net land use, land cover change emissions, estimating the ocean carbon uptake, which is pretty well constrained. We know how much CO2 is in the atmosphere, and then what's left is largely by land. Yep. Emissions, because it can't go anywhere else is the short answer. But so a bunch of it went into the atmosphere, but only some of it is still there. Yeah, so that 40% is that light blue bar. Yep. Okay, so this is it. This is reality. Like this is, for the most part, as best we know, what's happened to CO2 in the atmosphere. These are experiments now. So the C4 MIP class of experiments are these idealized experiments where they take all the Earth system models that want to play, and they're idealized because they add CO2 at 1% per year to the atmosphere, and they look at how much the land took up, how much the ocean took up, and then how much is left in the atmosphere, and what that does to temperature. So very similar to these experiments that are kind of more observationally based. These are idealized modeling experiments to try to figure out how much CO2 is left in the atmosphere. So they're 1% ramping CO2 per year, look at land and ocean carbon uptake, and then look at temperature change. And the interesting thing here, because it's a model, you can look at the fully coupled system, what happens when you put CO2 in the atmosphere, both radiatively and biogeochemically, but then you can also mask certain parts of that. So in the biogeochemically coupled simulation, that means the land and ocean see elevated CO2 in the atmosphere, but that CO2 in the atmosphere doesn't change temperature because you can turn off that feedback in the model. And then in a radiatively coupled model, it means that the land and the ocean see the temperature change, but they don't actually see the change in concentrations of CO2. So that's in a model how you can disentangle what's the radiative effect versus the biogeochemical effect, and then look at the integrated full effect. Guess what they saw? It's really tiny, sorry. This is in the fully coupled simulation, and the amount of the land atmosphere exchange. So this is the magnitude of the land carbon sink in these idealized experiments. So there's no, again, they're idealized. There's no land cover change. It's kind of a realistic amount of CO2 that's getting put into the atmosphere over these 140-year-long experiments. But what's this? What is this showing? Uncertainty in what? Yeah, so it's among models. Each line is a model. That's the part I kind of left out. So each line is a model, and that spread is uncertainty in the magnitude of the land carbon sink seen in this is for CMIP 6 models, but you see the same thing in CMIP 5 and C4 MIP before that. So this is kind of a persistent source of uncertainty in models. Most of that is because of the biogeochemistry. So this is the CO2 fertilization effects that land sees. And there's a little bit in the radiatively coupled simulation. So CO2 fertilization means that more CO2 in the atmosphere means that CO2 is basically plant food. So more CO2 in the atmosphere means that there's more CO2 for plants to absorb. So you get an increase in the land carbon sink. In the radiatively coupled simulation though, how come it goes down? This is this panel that I'm asking about now. Any ideas? If it gets warmer, why do you lose CO2 from why do you lose carbon dioxide from the biosphere? Yeah, so that's like, this is the planet inhaling, sucking up extra CO2, and this is the planet exhaling, blowing, oh, I lost, shoot. Spilled the beans. So this is the planet exhaling, and it's respiring more because temperatures are higher. Okay, so that net balance between the biosphere inhaling because of elevated CO2 and exhaling because of changes in temperature is kind of the ultimate thing that we're trying to figure out in land model biogeochemistry. Here's the full figure. That's why these plots were so kind of painfully small. So a few take-home points. Land biogeochemistry is hard, and it's because of that balance of understanding CO2 fertilization, which shows, oh man, so bad at this. This is the first, like, I've had to give 3 in-person talks in the last week, which is so exciting, but I'm just klutz at it now. Okay, so in the middle, CO2 fertilization is what's driving that spread in the biogeochemically coupled simulations. Warming is maybe not as interesting on land over this time scale, but there is some uncertainty there. Sorry if you're an ocean biogeochemist, it's really boring in the coupled system. It's all driven by physics, and so everyone knows what they're doing. That's what's shown on the bottom panel. That really tight spread means that there's very little uncertainty among models. And that either means that they're all doing the right thing for the right reasons, or all doing the wrong thing for the wrong reasons, but whatever it is, they're doing it consistently. And you're gonna hear more about ocean biogeochemistry on Thursday, I think. There's no spread in the ocean model. And it's largely because this ocean biogeochemistry there's a lot of fascinating biology that happens, but at this scale, looking at ocean atmosphere fluxes, it's all driven by physics. in land, it's all driven by biology, and biology is harder than physics. I know physicists think they're really smart, but it's not that hard. Biology is really hard, but it turns out to. And on the top plot, this is then the spread in temperature. And so there's a lot of spread in temperature, and it largely it's because of the airborne fraction of CO2 that's left in the atmosphere because of this uncertainty in land biogeochemistry. Okay, so it matters. This you can look up, the terrestrial carbon cycle or global carbon cycle. I clipped the figure to just focus on land. They're big pools, so that's what's shown like in things like soil and land biota. There's more carbon in soils depending on how deep you measure and who you're asking. There's 3 times more carbon in soils than there are in all of the biota and all of the atmosphere combined above ground. So it's a huge reservoir of carbon. So that's a big stock. Fluxes are also really big. These numbers seem small to me, but I'm not going to quibble. Something somewhere between 50 to 60 petagrams. I'm sorry, 100 to 120 petagrams of carbon per year go into the biosphere through GPP, that's gross primary productivity. And almost all of that comes back out every year. And so when we're looking at those wiggles on the CO2 trajectory that you see at Mauna Loa, what you're looking at is the residual. So it's what's left over from these huge fluxes. And so the residual is something like, 2 to 4 petagrams of carbon per year is what the land is soaking up. But that's behind this background of really big fluxes that are nearly unmeasurable with any of our current techniques. So there's huge pools, large fluxes, and the sink is the balance is this really small residual. And so part of why you see this really large uncertainty in land carbon models is because we're trying to balance all of these large fluxes, and then the net is kind of what's left over. So there's a lot of work to be done there. So how do we do that? This is kind of affectionately known the Brotherton diagram. Francis Brotherton was the president of UCAR and the director of NCAR from 1976 to 80, I think. And then he moved on to NASA and kind of was a huge leader in kind of having the vision to make Earth system models. Prior to this, land modeling was kind of seen as tangential. You just needed a black box that warmed up faster than the oceans, and you could get kind of circulation right. Adding biogeochemistry here with terrestrial ecosystems and marine biogeochemistry was kind of Francis Brotherton's one of his big contributions to Earth system science that lives on today. That then kind of launched this flying leap proposal, which Ines Fung and Peter Rayner and Pierre Friedlingstein, who did the Global Carbon Cycle project and also spent time here, proposed. And this is it was only 20 years ago, or 22 years ago. So they called it the flying leap, which launched C4MIP. And this idea of actually having prognostic CO2 in the atmosphere from Earth system models like CESM. That's matured into a figure like this, kind of showing that in the summary for policymakers from the IPCC report, that this link between CO2 emissions and temperature change is really critical. And it depends on the scenario that you choose and how much CO2 you emit, how much warming you're going to have. So these ideas of understanding the carbon cycle so that you can more accurately predict temperature changes is really critical for Earth system science. So both NCAR and CESM were key players in the development and concepts of creating the first fully coupled carbon cycle model that was kind of culminated with the Flying Leap experiment. And then since then, we've been leaders in other ways in adding coupled carbon nitrogen biogeochemistry, which came in CESM1, and we were the first Earth system model to include nitrogen in the carbon cycle predictions that were used in CMIP5. And then in CMIP6, we're also the first group to have explicit crop management in CESM2. So we're getting better and better at representing kind of complexity in terrestrial carbon cycling processes, going back to this kind of vision from the 1980s up until present day. So Gökhan probably showed this figure yesterday. I'm hoping he did. I think it's awesome for many ways, and it's tragic in others. And the biggest one is that it still has this separation between land and biogeochemistry. And those two things are in my mind, they're fundamentally related. And so the Bretherton diagram has this problem where terrestrial ecosystems, see if I can do this without changing things, terrestrial ecosystems are somehow decoupled from terrestrial energy and moisture budgets, which is not true. Like physiologically at a leaf level, water and CO2 and energy are exchanged simultaneously. And so separating those into a physical and biogeochemical processes that's not how it works in reality. And I think we need to kind of change our models and our thinking to integrate those better. Similarly, in CESM, this is how CESM works now. Land biogeochemistry is kind of seen as this tangential thing, and I'd argue that it's not. Partially that's because I'm biased. I'm a terrestrial biogeochemist, and it's like, no, we're important. But I think visually separating it like this is actually detrimental to the science, and we need to have better integration on those things. Okay, how do we do it? It's with a ton of people. I think the land model is unique in CESM in the breadth of contributors that we have working on this project. It's university collaborators from all over the world that are really putting their efforts into making biogeochemistry happen. So this is a slide that Donica put together a few years ago. And there's a lot of us. I think there's over 50 co-authors on the CLM5 documentation paper, which is just evidence of the huge community that's involved in working on this. I apologize, this figure looks a little futzy on the right side, but what we're trying to do is, and I'll kind of walk through this at a high level, is shown on the left side. So this cartoon is kind of trying to illustrate what it is, like all the processes, the stocks and the fluxes that we're trying to represent in both carbon and nitrogen, as well as disturbance, things like fire. We're doing that both above ground and thinking about plant canopies and below ground, thinking about soils and especially permafrost. And then the feedback below ground of how nitrogen availability affects plant growth, which affects leaf area index and other things. If you want a little more than the cartoon, Gordon wrote a book, which is awesome and very accessible, called Climate Change and Modeling the Terrestrial Ecosystem, which kind of lays out at a high level what we're trying to do, both in terms of the physics and the biogeochemistry and land modeling. And then if you really want to geek out, all of this stuff is up on GitHub. And so the escomp/CTSM website has a ton of resources, as well as just the code that goes into making this happen. So in a very tiny nutshell, here's what we're trying to do. At the highest level, we're trying to represent leaves. And so to represent we have to represent leaves because photosynthesis fundamentally takes place at a leaf level. Leaf level photosynthesis is governed by Farquhar, both Farquhar photosynthesis, as well as we're using the Medlins to model conductance. Previously we were using ball berries to model conductance. And those two are very similar, other than Medlin lets you change things a little bit by plant functional types. So we're fundamentally representing photosynthesis at a leaf level, photosynthesis and stomatal conductance at a leaf level, but then scaling that to the canopy. And so what happens at a leaf is great, and that's something you can actually go out there with a light core and measure. But you can't, but we have to scale it to a canopy level. And so we're doing that with this two-stream approximation of sunlit and shaded leaves, which is a pretty simplistic way of representing the heterogeneity that's in a canopy. I'd love to talk more about that kind of canopy scaling and ideas that especially Gordon has about doing this with a multi-layer canopy. But we're taking leaf level processes, scaling them to the canopy, and then that's giving us kind of an ecosystem level of gross primary productivity, or GPP. So that's the inhale of the carbon cycle. Then those plants have to allocate that carbon to different tissues. Some of it goes back to autotrophic respiration. So that's part of the exhale is plants suck up CO2, but then they also release it. If you don't learn anything today, it's really important. One of the biggest misconceptions in science is that plants don't respire. They absolutely respire. They have mitochondria in their cells. And if you don't know what any of those words mean, I'm sorry, but it's really important to know that plants also respire. And so they do that in the model. And that there was a question earlier about the tight coupling between, interannual variability. And right now in CLM5, the coupling between GPP and autotrophic respiration is too tight, and that's what's giving us a really low interannual variability in our carbon cycle. So after plants blow off some of the CO2 they just took up for their respiratory processes, then they allocate the rest of it to leaves, to wood, and to roots. And so they build tissues that stick around for various amounts of time. We have to think about phenology, which is really tricky in a coupled sense. When you don't care about what plants do, you just tell the trees when to have leaves and they grow fine, and you don't worry about the carbon cycle. But when you do care about the carbon cycle, you have to let the plants figure out when to put those leaves on and when to drop them. And that's really tricky to try to do globally. So mortality and turnover, once when a tree drops in a forest, does anyone hear it? I don't know, definitely not the leaves. So when those leaves drop in the forest, then they start to decompose, and that kind of completes, kumbaya, the circle of life, and CO2 goes back into the atmosphere. So all of this happens in CLM, and it happens differently. So I don't know what the tutorial is today, but if you are a land person, here's just like the quick page, and these slides are available for some of the fluxes that you might be interested in. So GPP is gross primary productivity. Autotrophic respiration, we abbreviate as AR. You can subtract GPP from autotrophic respiration, you get NPP. That's the amount of kind of biomass that then sits, is what the plants have to build biomass, especially leaf area index, which we abbreviate with ELAI, which is exposed leaf area index. And then the downstream parts are heterotrophic respiration, NEP, NEE, and NBP, which is just a whole mess of letters, and it's just subtracting different parts of the fluxes out. So at the end of the day, NBP is net biosphere production, which is net ecosystem exchange minus land use change minus harvest fluxes from the from both wood and from crops, and that's the CO2 that gives you that increasing CO2 in the atmosphere. It gets tricky though, because LAI affects GPP through this canopy scaling process, and heterotrophic respiration also affects gross primary productivity through nitrogen availability via LAI. So one of the hard parts about representing terrestrial biogeochemistry is all of these feedbacks where you think you're changing something downstream in the land model that's not going to affect LAI, because if it feeds back on LAI, all of a sudden you've changed the whole carbon cycle. And so spinning up the model and getting kind of a stable carbon cycle to run with without breaking everything is one of the tricks. And that's partially because of this feedback of LAI on GPP. And it's also because of nitrogen feedbacks and how nitrogen decomposition processes in the soil that affect nitrogen availability that then feed back on GPP. And then it cascades all the way down this kind of trunk. Did Dave show this in his talk? Cool. This figure is really complicated and there's so much good stuff in it. So I want you to focus on the bottom part here. And so all of these processes happen at a PFT level, or a plant functional type level. And so that's just the simplistic way that land models use to recognize that an evergreen tree is different than a deciduous tree, which are different in tropical versus temperate areas, and they're all different than grasses. And so in CLM, we have 16 different plant functional types, as well as a number of crop functional types that I'll talk kind of briefly about, because I don't think you guys get an introduction to the crop model in this tutorial. So we think about natural vegetation, which is all on its own column. It shares a soil column. So within the subgrid architecture of CLM, there's soils, and on top of those soils are all the native vegetation. And then a separate column is where all the crops are, and each crop is on its own column for reasons that I'll talk about in a minute. So ag and CLM, what crops do you think we should try to represent? Corn, corn's king. What else? What? Rice, soy, wheat. You guys are nailing it. Any others? What? Oh, sorry, I can't hear you still. Mango, that would be awesome. We don't do tree crops. That's a huge problem though. We don't do tree crops and we would like to. Yeah, so these are all annuals. A couple more, cotton and sugarcane so both corn and soy have temperate and tropical varieties. So we do think about varieties for those two. And then, so anyway, there's 8 total. With the addition of now, we have a winter wheat parameterization that seems to be working, as well as 2 biofuels crops, so switchgrass and miscanthus. Yeah, there is an oil palm parameterization that has been made for CLM 4.5, and it never has been integrated into CLM. That would be a really nice one to have. Yeah, it's there, just not in the main code base. Yeah, which would also give us a tree crop, I guess. So agriculture and CLM is I think one of the exciting things that we do, and a huge growth area. So we only have, I guess, 8 plus 2 more, and winter wheat is 11 crops that we're really representing in the model now. But we can hold as many as, I think it's 78 crop functional types. So if you really think that, I don't know, barley is different than wheat and you want to represent barley in the model, or you want to represent mangoes or coffee or chocolate or anything else that's delicious and important to think about, there's room in the model for it in the architecture. We just don't have the parameterizations for it right now. So on top of it, we also think about management. And so we can fertilize and irrigate. And so on top of having corn, you can have fertilized corn or irrigated and fertilized corn. So there's a number of ways that you can kind of mix and match these things. And that crop management is really important to the carbon cycle, and some work that our colleague Donica Lombardozzi has been doing. Okay, so where do these parameterizations come from? This is like going to be super fast. This is kind of where the magic gets made. The first thing is from laboratory incubations, and in a kind of fundamental understanding of plant physiological processes. And so, like I said, the Farquhar photosynthesis model recognizes that photosynthesis is co-limited by light, energy, and the export of sugars. So we have all 3 of those in the Farquhar photosynthesis model. We also use empirical relationships. So the empirical relationships, especially from big databases like TRI, where you can measure something that's relatively easy to measure, like foliar nitrogen concentrations, and then understand how foliar nitrogen scales with, for example, dark respiration or BC max or J max or anything else. You can look at other things like specific leaf area and leaf turnover times. So there's a number of ways that you can use empirical relationships from the literature, and especially large databases like TRI, to do this kind of stuff. There's other databases like FRED, which is an awesome name for a database, which is the fine root. It's about roots. We don't use FRED very often, and I think those are opportunities that we could think about building on. And this got formatted kind of funny in moving onto Google, but we also use optimality theory quite a bit, which is kind of recognizing that plants, we assume that plants are rational actors that try to maximize their fitness on average. And so that's represented in components of the model like FUN, which stands for the fixation and uptake of nitrogen, which allows plants to kind of look for the cheapest source of nitrogen in the soils, and that also makes them pay a carbon cost to uptake that nitrogen, as well as the Luna module, which is used in photosynthesis and optimizes BC max and J max based on temperature and CO2 concentrations in the atmosphere. So where do these parameterizations come from? It's a variety of sources, and some of them are very data constrained, others are not. Many of the PFTs that we try to represent have PFT specific parameterizations. Some of those, it makes a lot of sense. We know that needle leaf evergreen trees hold onto their needles a lot longer than a tropical deciduous tree does, but we don't know exactly how long they do. And so we kind of come up with logical parameterizations for these things. And other times there's room in the code for PFT specific parameterizations, but if we don't know if an evergreen tree or a grass are different, then we just leave them all the same to try to reduce the guesswork, I guess, in all of this. But the parameterizations of all aspects of the carbon cycle, I would say, are highly uncertain, and one of the big challenges that we have. So how am I doing on time? Okay, good. What's that? Okay, cool. So I think Dave hopefully showed this figure too. It gives me a lot of hope looking at this, just in how new a lot of this science is. So a lot of the science has started really in the 2000s when we added a carbon cycle and dynamic vegetation models to CLM. More recently, we added things like nutrients, land cover change, which Peter has been instrumental in, as well as crops. And so, on the top half of this diagram, a lot of the science that we're trying to do is relatively new. And so it's okay that we're not great at it. So how are we doing? Some of this is kind of rah-rah, like we're doing better than we were before. This is from Gokon's 2020 James paper, and it's the sign of the land carbon sink. So this is huge in CESM1. The land was not a sink of CO2 from the atmosphere. It was always a source all throughout the 20th century, which is not good. That's what's shown in the brown online here. And in CESM2, we've reversed that so that over the satellite record, or over the GCP era from 1960, over the measurement record from 1960 through present day, we're capturing the magnitude of the land carbon sink surprisingly well, which is great. Whether or not we're getting that land carbon sink in the right place or for the right reasons is a different question, but we're at least within the, we have the right sign on this curve, which is good. Objectively, things are better. Did Dave talk about ILAM at all? Oh my gosh, he didn't talk about ILAM. Okay. ILAM has radically changed how we approach land modeling and land modeling evaluation. So it's the International Land Model Benchmarking Package. And it's kind of this collection of data sets that we can use to evaluate where we've made improvements in the model, where things have gotten worse. The kind of take home here is that everything for CESM2, which are the right three most columns, are green for the most part, are green, which means they're better than CESM1. So in the coupled model, we're having a better carbon cycle, which is shown on the top set of plots. We have a better hydrological cycle, largely, which is shown in the blue set of plots. We have better radiation and energy, which is partially because of land, but also because of CAM. Okay, so we're getting better. And for purposes here, in terms of ecosystems and carbon cycle, we're doing better on everything, ironically, except for soil carbon. And we were the worst model in CMIP 5 for carbon cycle, and we got even worse for CMIP 6, which is astounding because we added permafrost. But this is CAM's fault because it snows too much in the Arctic in CAM, which means it's covered with snow all the time, which means you can't grow plants. And if you can't grow plants, you can't have carbon in the soil, which gives us red bars for this. So it's CAM's fault. It's not CLM's fault on this one. They're always blaming us for stuff. I'm gonna show this video because it's cool, if it works. This is the flying leap. So this is for CESM2, the fully coupled model where we're actually trying to prognose CO2 in the atmosphere. And what you're seeing here with the kind of lightening and darkening of the shades is summer and winter. So when the colors get cooler or more blue, that's summertime and the land inhaling. When the colors get warmer, white and eventually red, that's wintertime and the land surface exhaling on net to the atmosphere. You can also see point sources of CO2 here. So you can see kind of industrial areas of North America, Europe, and Asia, which is the source of anthropogenic CO2 in the atmosphere. And to me, this is so exciting because all of it is coming directly from the model. We're giving CESM CO2 to the atmosphere in these places, but then it's figuring out the balance of where all that CO2 goes. And what you're seeing in the little line graph on the right is kind of similar to the Mauna Loa curve, but I drew it for Niwot Ridge, which is just up the hill and the second longest CO2 record after Mauna Loa. Yeah, go ahead. Sure. Oh, sorry. Yeah, the dot is Niwot Ridge, which is where these observations are taken from. And actually, I'm not showing the observations here, but there's about a 15 parts per million CO2 offset. So we're about 15 parts per million high in CESM2, but that high bias is consistent throughout the historical record, and it started at initialization. So in 1850, when we initialized the ocean model and the land model and the atmosphere all together, we started, our equilibrium was about 15 ppm high, and we maintained that. So there is a bias we're having, but we're kind of, aside from that bias though, we're able to calculate this amazingly well, which is so exciting. Now I can't figure out how to get back to my talk. Can you guys help me? Maybe I can do it. Yes. Okay. I'm going to skip the last few of these and talk about where we're heading next. And so there's kind of, the big question is really how will ecosystems function, and how will ecosystem function and vulnerabilities transform under climate change? And we're doing that in kind of three ways. One is with the representative hill slope model, which I'm not going to go into now because I think Dave talked about. And that feedback of hydrology to biogeochemistry is really important. And so that's something that we're actively trying to work on. The next way we're trying to do that is with fates. And I think Jackie's going to talk to you about fates in one of the application talks later on this week. But it fundamentally changes how we think about this tiling structure of PFTs, of having, broadleaf and needle-leaf trees that are evergreen or deciduous and grasses, and instead thinking about tiling the land surface in terms of this time since disturbance. So you can have patches on the landscape that were freshly disturbed, they just burned in 2021, and then patches that are older that might be 10, 15, 30 years post disturbance. Then within that, there are cohorts of plants. So cohorts define kind of the size of the plant. And then there are PFTs. And so, you can, and the interesting thing here is that in fully functional and fully transient fates, we're not telling any plant where to grow. We're just, we're giving the model, here are all the plants that could be growing, and the model has to figure out where they actually grow and when they do better in terms of the disturbance regime that's happening in the place that they're growing. And so you can have tall PFTs and short PFTs within the same patch. And so it really transforms the way that we think about ecosystem demography, and kind of more realistically representing the time since disturbance and how that affects carbon cycle on the land surface. So there's a huge team working on fates. And then the last way is below ground and thinking about functional traits in the carbon cycle. So on land, we talked some about plant functional traits and how those leaf traits are really important for kind of controlling the green part of the carbon cycle. Ocean biogeochemistry thinks about plankton functional traits, which is awesome. That's another PFT, but that those plankton functional traits are really important for controlling the blue part of the carbon cycle. And so some of the work that I'm heading up is thinking about microbes and how biodiversity below ground is really important for controlling the brown part of the carbon cycle and patterns of decomposition below ground. And so, we consider biology above ground and at sea, and how about in the world beneath our feet. So that model is called the Mimics model. And that's in the CESM 2, or it's in CLM5 code base now. But we're having some spin up problems that we're working on with the ocean biogeochemistry team to work on spinning up, using some of the tools that they use to spin up the ocean model to also spin up the land model. So I'll leave with that and kind of the future directions of where we think we'd like to go. Hopefully I'm okay on time. Thanks.
So we do have a bit of time for a couple of questions.
Okay,
I'm also going to say that we do tomorrow I'm giving a land cover change talk, and
right.
then Jackie follows up with a ecosystem demography and fire talk. So that's what happens when you leave a land person in charge of the CSM tutorial. But there will be continued discussion on this. But if you have questions for Will now, please raise your hand and we'll bring the mics around. And sorry, Ryan or Elizabeth, can we bring mics around? Thanks.
One here and one here.
Hi, thank you for the wonderful overview of the biogeochemistry. My question is, how are human decision-making incorporated into CLM? When to plant, what to plant, that kind of stuff.
So right now so you're asking about the crop model specifically. So the crop model uses a growing degree day threshold and kind of planting windows for when to plant. I'm kind of pausing because this is all changing. Right now we're using a crop model that's based on agro-IBIS, and we're switching hopefully before too long to something that's based on AppSim. And I don't know the difference. I'm not a crop modeler, so I don't know the difference between those, but it kind of uses this growing degree day threshold for when plants actually grow within a window, a planting window. But it's all temperature driven. And it's not moisture driven at all. And so we don't have any feedbacks for if it's too wet to go plant in your field and you delay planting. That's not in there. We also don't have, one of the problems we have now with CLM5 is that across India, it's warm. And so farmers in the model go plant during the dry season and all the crops die. So yields in India look terrible right now. And it's because we're missing the moisture feedbacks that we really need to be representing. So yeah, it's temperature and growing degree days though.
Yeah. So we do have a postdoc working on planting calendars. So you can have an alternative instead of using the default growing degree days from the beginning of the growing season. They can actually change the window. So that will be something that Sam Rabbit.
Yeah. Sam's work is super cool. The hard part too is then how do those planting windows change with climate change? So Sam's work will let us kind of do a good job now. And I don't know how that can modify in the future. We also don't do other, like there's a lot of other things that we'd like to add to the crop model that aren't there. Like we'd love to have multiple sowing date, like multiple harvests in a year, and you know, crop rotations and cover crops and all that kind of great stuff. We're not there yet. So it's not an ag model, it's a crop model within an earth system model.
I was going to say the fertilizer is sort of pretty much set at the beginning of the growing season, and the amounts are prescriptive. So we have
Yep.
time series for each grid cell, for each crop type, how much fertilizer gets applied, but we don't say that this much fertilizer gets applied at this day and this day and this day. It's more just like we have a general, this is how much is the annual amount of fertilizer, and it'll be applied in an optimal manner. Same thing with irrigation, it's on demand rather than prescriptive.
Yeah, the irrigation's more on demand.
Hi.
Yeah.
Thank you for your great talk. I really enjoy your talk.
Thanks.
And I was wondering, is the biogeochemistry model interact with the COM model? Because it seems like the CO2 fluxes will kind of affect your transpiration. So I'm just wondering if you will have any interaction with.
No,
the
they're
model.
coupled
Yeah.
together. And so, yeah, that's one of the improvements we made in CLM5 was that previously water and carbon cycles were somewhat decoupled because of nitrogen feedbacks. I can talk for a long time about this later, but that's changed in CLM5 so that those water and carbon cycle fluxes are coupled. If you're running a biogeochemistry run, if you're running an SP simulation with satellite phenology, we don't really care about the carbon flux. The carbon fluxes happen, but they don't affect anything else.
Okay, thank you.
Getting your exercise, Ryan.
Hello, thank you very much for the presentation. It was very interesting. It's very exciting to see the development in the land component and how we extend the capabilities, for example, to better represent the different PFTs with their parameterization, and also now vegetation demographic and so on. And we also go now in more details by accounting microbial activity. So I was wondering, we know that ecosystems are a very complex system where everything is connected, including maybe insects and birds. And we see that climate change affects their chances of surviving and also their shifts in their position, geographical position.
Mm-hmm.
So I'm wondering if it does play a role, a sensible role for climate applications on distribution of plants, for example, and so on. And if yes, is this something like we intend to implement in the model or not really?
It ends up being kind of a technical problem. So like this, to me, what your question is about is how you move things across grid cells. And so the ocean and atmosphere models have to think about that a lot. The land model is basically every grid cell is its own thing, and it doesn't have to communicate with other grid cells. And so it means to have insect pests or seeds or birds moving across grid cells means that grid cells have to know where they are in relation to other grid cells within the coupler, which is something that we don't do right now. So it's mostly a software engineering problem that would open up those kind of scientific questions.
But do you think they have a sensible impact on, time scale of projection?
For sure, especially for plants. Like if you want to think about, the boreal Arctic interface and trees moving into the Arctic under climate change, you have to, that rate of change is dependent on how quickly the seeds can get there and then their survival. But some of it is a dispersal problem.
Okay, cool. Thanks.
Yeah.
And I think this is something that Gokhan brought up, is like some things we physically represent, sometimes we have to parameterize. So if you can get those emergent properties captured in the model, it might not necessarily explicitly, you've got each insect coming through and like the impact on the trees. We might not be able to actually model that, but we can model certain elements such as the impact that would have on the trees and also on their distribution. Right.
Yeah. One of the coolest applications I've seen with CESM was a woman who looked at wheat rust and the which is a fungal pest that affects wheat, and the distribution of wheat rust and how it could spread around the world using CESM. It was awesome. It was so cool. But it mostly used CAM to kind of with this probability distribution function to say like, okay, if you have wheat rust here, where's it going to spread to, kind of thing. It was really neat. Yeah, go ahead.
Oh, sorry, I'm actually going to call time just because it's 10 o'clock.
Oh.
We're actually
Okay.
15 minutes behind on the schedule.
Okay.
So sorry.
I'm going to be here all day,
but-
so if folks
But
want
we'll
to
be
grab
here
me.
so we can have discussions and just so we can get a break before the next talk. We're going to go break now and we'll come back at 10:15. I apologize. Like, I know your question's important and I don't mean to shut you down, but we really are 15 minutes behind schedule already.
Okay.
Thanks, Will. I really appreciate it. And yeah, we'll see you all back here at 15:15 for Clara's talk, which is going to be on looking at ensembles and looking at internal variability of models. So thanks.
Cool. Thanks.